So, ladies and gentlemen, you are all welcome, and we are now going to start with the general revision. And I hope this of our tutorial. Okay, let me share the link first. Let me share the link. Okay, so good evening. You are all welcome. So I think what you are going to do, I will start by asking you where do you have problem, or you can just ask questions. Then before we start answering first question paper. So please ask your question. Okay, yeah, Maria Memunat Muhammad. Yeah, you can ask question. Where did you have problem? Before we proceed. So you can ask question. So you are all welcome. I said you can ask your question. Ask question. Oh, I should just answer what I have. Okay, good evening. You are all welcome. Truly, sir, I don't understand this course at all. Maria Mario, you said that you didn't understand this course. So it means that you didn't understand everything in this course. So I think I told you last week that I'm going to make, last 2 weeks that I'm going to make a brief description of this course for 3 hours. So I think I made like, if I can remember, I have a video of close to 2 hours that I try to summarize the course. So I will advise you to go and watch that video. It is already there, so I send it there. Okay, module 1, oxidation of saturated and unsaturated fatty acids. So let me start with Efunania Margaret question. So actually, when we say saturated fatty acid, ladies and gentlemen, let me just say it very clear. Saturated fatty acids are those fatty acids that in their carbon chain, they all have single bond. They all have single bond in between them. and that is what you call saturated fatty acids. And then we have unsaturated fatty acids. So when we said unsaturated fatty acids, they are the fatty acids that have at least one or two double bond in them. So those kinds of fatty acids are called unsaturated fatty acids. So unsaturated fatty acids, simply it means that they have at least one or two double bond or triple bond in them. And most of that unsaturated fatty acid, we don't actually have them in human. Most of those unsaturated fatty no yeah, most of them, we don't have them, or we cannot actually synthesize them. We usually get them from external source. So we can even say that they are essential because we cannot synthesize them in our body. And one very interesting thing that I wanted to say here in addition, because I saw some of the question papers that actually ask some of the unsaturated fatty acid that are of physiological importance, like omega-6 fatty acid and omega-3 fatty acids. They are also examples of unsaturated fatty acid, and their main importance in the body, they are actually trying to uh, help us in so many physiological process. Number one, they can serve as an antioxidant, that is, they prevent oxidative stress. And then number two, they increase our level of fertility, because in fact some people are infertile because of the deficiency of this omega-3 and omega-6 fatty acids. They also actually improve the quality of eggs from women. It also increase the quality and the quantity of sperm in men. So therefore they increase fertility. So that is in the case of saturated fatty acids. So what other questions do you have? Or should I start answering from the question paper? Should I start answering from the question paper? Should I start answering from the question paper? I'm waiting for your response. Yeah, I share it already. I think I share it more than 5 times. So you check your WhatsApp group. And even you that you are asking, Riri, you are already members to the channel, so you can watch the video. I already uploaded it. It's only those that did not join the membership, that is actually the one that are going to have problem with those two videos lecture. Sorry, 2 hours lectures. And one very interesting thing, while answering or while summarizing the course, I try as much as possible to answer it, and also at the same time solving the question paper at the same time. Queen, you are saying that area of concentration. So let me just give you this hint so that at least you understand one thing that is actually associated with this course. Number one, I can't say this is the area of concentration, because the reason, I'm not the only one that is taking the course. So we submit all our question papers. So they will select the one that they feel they will actually ask you. So it is actually a general something. Okay, using specific example, briefly highlight the relevance of biochemistry to understand key concepts in nursing science. Yeah, I think I answered questions like this last time. Biochemistry highlights or is actually very important in nursing, in drug treatment, in drug development, disease diagnosis, and treatment. So we use biochemistry, or biochemistry is important in nursing, in drug discovery, disease diagnosis, and disease treatment. So these are the importance of biochemistry in nursing science. So you said the relevance of biochemistry to understand key concepts in nursing science. So that is in the management of disease, in the treatment of the disease. Like for example, you can use nutrient to treat disease and so many other disease condition. And I think, okay, let me answer this question. Let's start from the question paper. I think it will be the best. Question 1 says highlight the relevance of biochemistry to drug discovery using a specific example. So let me just give this example. Uh, Drug discovery, when we say drug discovery, generally in drug discovery, we have we must have an enzymes or protein, because I have said this in the video summary that most of the drugs, they are either protein inhibitors or protein activators. They are protein inhibitors or protein activators. So let me just give this example. If you now take cyclooxygenase with a specific example, you can mention many. So cyclooxygenase is an example of enzymes that is actually involved in the synthesis or in the production of prostaglandins. So prostaglandins, they are lipid molecules that they usually associated with headache and stress. So whenever we have high concentration or high amount of prostaglandins in our body, it is an indication that we either have headache or stress. So therefore, if we have that situation, what are we going to do? So you can actually look at the enzyme that is responsible for producing those prostaglandins like cyclooxygenase. The name of the enzyme is cyclooxygenase. So you can try to identify a molecules that can serve as an inhibitor or that can pit to the active site of that particular enzymes and stop its activity or to decrease its activity. So any molecules that you design or that you use to block the activity of that cyclooxygenase, it means that the molecule can serve as a drugs. So that is one of the important things that I wanted you to understand. And then in addition, we can also look at the drug discovery from infectious point of view, that is from the infectious agent. Plasmodium falciparum is a parasite that causes malaria in humans. So if you can be able to identify a protein that is important for the survival and the growth of the parasite in humans, like for example in the red blood cells or in the liver, what are you going to do? So once you identify the proteins, then you can try to identify a molecules. Although in the process of identifying the molecules, we have a series of steps and different set of technologies. So when you use a technology to identify a molecules that can actually bind to the protein that is important for the survival of the Plasmodium falciparum, it means that you discover a drug. So that is the importance of biochemistry in drug discovery. I think this one is clear. Do you understand the first question, that is 1A, the relevance of biochemistry in drug discovery using a specific example? So like the first one I said, cyclooxygenase produce prostaglandins that are able to identify a molecules that can actually inhibit the activity of the cyclooxygenase. It means that that molecules can serve as a drugs. So that is what I said. And another example, currently I'm working on a research where I was able to identify a protein called sortilin, and the sortilin is a protein that's important for the invasion of Plasmodium falciparum into the red blood cell. So it means that if you are able to identify a molecules that can block the activity of the sortilin protein, it means that the Plasmodium falciparum parasite cannot get into the red blood cell. So it means that the molecules that can block the activity of the sortilin protein can serve as a drugs for the treatment of malaria. So in simple term, biochemistry is important in drug discovery by identifying a molecules which can serve as an inhibitor or activator of an enzymes or a protein. Someone is asking that it is essential fatty acids that can also be considered or can also. Okay, Rukaya, thank you very much. Wow, mashallah. Yes, thank you very much. So that is actually for that. So someone is also asking, is it essential fatty acid that also be considered called unsaturated fatty acids? When we said essential fatty acid, they are essential because we cannot synthesize them in our body. So we get them from the diet. That is why they are called essential fatty acid. But unsaturated fatty acids, they are fatty acids that have at least two or three uh that have at least two or sorry that have at least one or two double or triple bond in them. At least if you look at their chain, you will see that they have at least one double bond or triple bond in them. So that is unsaturated fatty acids. And of course, most of those unsaturated fatty acids, we cannot synthesize them. So most of them, they are essential because we cannot synthesize them. That is why we can call them essential fatty acids. Is that clear? Or double? Is that okay? I will advise some of you that actually didn't join the YouTube community to try as much as possible to join the YouTube community. As I said, very soon, immediately after you finish your exam, because this time around, if you realize that we are very late, most especially for this biochemistry lecture. So I think we are going to be fixing at least 2 days in a week so that we will be discussing all the biochemistry that we are going to do physiology and microbiology in your next semester. So that is why I always advise you to try to join the YouTube community. So I answered that question. So let's move to question 1b. Highlight how glycoproteins are involved in ABO blood group system. So what I want to say now, first, in ABO blood group system, remember we have different blood group types. We have blood group A, we have blood group B, we have blood group uh, O. So an individual, the sugars that are found on the or the glycoproteins that are found on the ABO blood group system, we have number one, we have uh, galactose and acyl galactosamine. These are the sugars that are found on the surface of the red blood cells, and that is why that is why we actually have different blood group types. So, like for example, an individual with blood group A is having blood group A because of the presence of N-acetyl galactosamine. So that is the glycoprotein that is found on the red blood cell. While an individual with blood group B is because of the presence of galactose, while an individual with blood group AB is because of they have N-acetyl galactosamine and galactose, while an individual is having blood group O because the individual doesn't have N-acetyl galactosamine and and galactose, N-acetyl galactosamine and galactose. So an individual is having blood group O because of the absence of these two sugars. Please, I joined the channel membership late. Please, the 2 hours revision lecture, how can I get it? Yeah, I will send it to I will resend it to the group after we finish this. So I will resend it to the group. Okay, someone is asking, can you please type these glycoproteins here? Yeah, let me type it. As I said, we have N-acetyl galactosamine. So N-acetyl galactosamine is found on the surface of red blood cell of an individual with blood group A. So I'm is here. So N-acetyl galactosamine on blood group A individual, while galactose on the surface of individual with blood group B, that is galactose. And then for an individual with blood group AB, it means that the individual is having both N-acyl galactosamine and galactose. While an individual with blood group B, it means that the individual, it doesn't have actually N-acyl galactosamine and the galactose. So then now, let's answer question 1C. What is the basis for using serum aspartate transaminase and alanine transaminase as a markers of liver damage in humans? So this is actually the one of the area of biochemistry in disease diagnosis. Are you not seeing? I am sharing a slide. Are you not seeing the slide now? Can you see what I'm sharing? I said, are you seeing what I'm sharing? Okay, so let me share it again. Sorry. I thought that it is showing, so let me share it again. So I'm sharing it again. So can you see it now? Can you see the questions? Can you see the questions now? Okay. So look at question 1C. Question 1C says, what is the basis of using serum aspartate transaminase and alanine transaminase as a markers of liver damage in humans? So now what I wanted to say, in a serum or in a blood, there is a normal range for alanine and aspartate transaminase enzymes. And these enzymes are always localized, or they are always present in the liver. So when there is a liver damage, when there is a necrosis, when there is a serious damage in the liver, the content of the liver will be released into the blood. So it means that whenever you take the blood or you take the serum sample and you realize that the concentration or the amount of aspartate and alanine transaminase is high, so that means that there is a liver damage. So that is the diagnosis. It means that if you're supposed to have the enzymes in the liver, but now you check in the serum that the amount of those enzymes, aspartates and alanine transaminase, and you now realize that the amount of that enzymes in the serum is high, so it means that the liver is damaged. So that is a strong indication that the liver is having problem. You realize that you have a content that's supposed to be in one particular organ, and now you now find it in the blood, so it means the organ is having problem. Is that clear? Do you understand now? So the enzymes used as a marker for the diagnosis of liver damage because they are only supposed to be in the liver. So when the liver is damaged, then part of the content that will be released, it is these two enzymes. Okay. So the next thing that we are going to look is question 1D. Question 1D, I hope that question 1C is clear. So question 1D, what is the relevance of vitamin or B vitamins and their derivatives to catalytic reaction in our body system. So please read out the question again. I can see it clearly. Yeah, I'm reading the question 1D now. Question 1D says, what is the relevance of B vitamins and their derivatives to catalytic reactions in our body systems? So remember in one of the in the revision that I made on the last video on enzymology, I made mention that enzymes are most of them they are produced as an inactive form. So the inactive form of an enzymes is called apoenzymes. So those enzymes they require cofactors or coenzymes for them to become active. So some of these coenzymes that are required for the enzymes to become active, and remember when we said enzymes, there are molecules that generally speed the rates of reactions in our body system. So if the enzyme is inactive, it means that there is no reaction. And if the enzymes are active, it means there is a reaction. So what makes the enzymes to become active are what make the enzymes to become active are the coenzymes. And most of the coenzymes are vitamin B complex, especially vitamin B1, vitamin B3, and vitamin B6, and also vitamin B12. So vitamin B1, vitamin B3, vitamin B6, and vitamin B12, they are the vitamin B complex members that are involved in activating the enzymes. So the answer to this, vitamin B12s, they are coenzymes that are required for the enzymes for them to become active. Most of the enzymes, they are synthesized as an APO enzymes. Let me write it here, APO enzymes. So when we said APO enzyme, it means they are inactive enzymes. So they need to be activated by a coenzymes or cofactor. So vitamin B12, as I said, or vitamin V members or the B vitamins, they are called coenzymes. Let me also write it here. They are called coenzymes and they are needed for the APO enzyme. So when you have APO enzymes plus coenzymes plus cofactors, that will give you the active enzymes. And the active enzymes is called holoenzyme. I also wrote it on the comment section, on the comment section. So we have alpha enzymes inactive, holoenzyme is active enzyme, and these alpha enzymes require coenzymes and cofactors, and most of the coenzymes are the B vitamins. So that is why the enzymes require these B vitamins for the catalytic reactions to take place in our body. Is that clear, ladies and gentlemen? Is that clear? Is that clear? I'm waiting for your response. Do you understand? Okay, excellent. So now, let me also. I have seen that so many of you have didn't join, are yet to join the YouTube. Excellent, Abdul Mumin Tahira. Welcome. Thank you for joining the YouTube. So as I said, try to join. In fact, there are so many videos that you cannot see them, you cannot watch them if you are not a member. So that is why I advise you to try as much as possible to join. Okay, Echekuchu promise. This video will still remain on the YouTube. I don't want us to keep repeating, keep repeating. You can always come back and listen to the lecture. But let me repeat it briefly. I said most of the enzymes they are inactive. They require coenzymes or cofactors to become active. So most of the coenzymes that are required by the enzymes for them to become active, they are the members of vitamin B complex. So vitamin V or V vitamins and their derivatives are required by the enzymes for them to become active. And if the enzymes are active, it means that the catalysis will take place. And if the enzymes are inactive, it means that there is no catalysis in our body. So the enzymes, which is actually inactive form, the inactive form of the enzymes is called APO enzymes, and they require these B vitamins for them to become active. So that is actually the answer to question 1D. So let's move to question 2A. The question 2A says, describe the functions of the following organelles. So the first organelle is endoplasmic reticulum. So who can tell me what is the functions of endoplasmic reticulum in a cell? Let me get the answer on the comment. What is the functions of endoplasmic reticulum of a cell? Akinabo Oluwasion, thank you very much. Welcome to the YouTube channel. So you can now have access to all of our videos. And I promise that we are going to take in the very few weeks after writing your exam, we are going to continue. So you see, some of you are writing that endoplasmic reticulum is used for the protein synthesis, and it's wrong. That is not the correct answer. That is not the correct answer. And so, no. Rough endoplasmic reticulum, or the endoplasmic reticulum, is not involved in protein synthesis. What is the answer? Who can tell me the functions of endoplasmic reticulum? No, it is not involved in the storage of protein, degradation of worn out tissue. Do you mean tissue? No. Seriously, ladies and gentlemen, you didn't read very well. What is the synthesis of drugs? No. Lipid synthesis. Mm, synthesis of lipids. Protein synthesis and degradation of worn out organelles. Yeah. So let me just actually give you a brief site for drug metabolism. Yeah, because remember, yes, so you see the answers are here. Penum ilia, you say site for drugs metabolism. Because we have two types of we have two types of endoplasmic reticulum. So we have smooth endoplasmic reticulum. So in the smooth endoplasmic reticulum, it is where the drugs are metabolized. And also the packaging and the transportation of proteins is carried out by the rough endoplasmic reticulum. Yes, also storage separate and serve as a cell transport system. Yes, Otata patient, you are also correct. So remember, endoplasmic reticulum is divided into two. So smooth endoplasmic reticulum is held in drugs metabolism. That is one. And then second, the rough endoplasmic reticulum is held is aids in protein packaging and the transportation of the synthesized protein. Then who can tell me what is the function? So we are done with the function of endoplasmic reticulum. Then the next one is lysosome. Okay, endoplasmic reticulum has both smooth and rough. The rough is for detoxification of harmful substances in the hepatic. Yeah. So now, who can tell me what is the functions of lysosome? So we are now done with the endoplasmic reticulum. Endoplasmic reticulum, as I said, is divided into two. So smooth endoplasmic reticulum is for drugs metabolism. The ROF is for transport and the packaging of the synthesized protein. So then what is the functions of the lysosomes? The functions of the lysosome, who can tell me? Lysosomes, also known as a excellent suicidal pack. It helps to engulf toxic substances or even microorganisms. In fact, when microorganisms are engulfed by the immune cells, it is these lysosomes, because in the lysosomes we have an enzyme called lysozymes. So it is degraded by the lysosomes. So the bacteria and the foreign substances that can cause damage in our cells are digested and degraded by the lysosomes. Don't forget to join the YouTube community. There are so many videos, especially the summarized video on biochemistry. It's only if you are a member that you can watch them. And don't forget that immediately, as I said, after like 2 weeks of your exam, then we are going to continue with the normal biochemistry courses and physiology for your first semester. So we are going to even fix like 1 hour in a week, or 3 hours at least. Let's say 3 days, and each day we are going to have 1 hour to have a general class of all these courses. It might not be necessary that I will be among your tutor, but I will try as much as possible to help and assist you to start doing these courses on time, so that at least you will not have problem. You will not start doing this kind of die-minute jacking. So now, after endoplasmic reticulum, lysosomes, then the next one is ribosomes. What is the functions of ribosomes? Okay, Osuyumi, I said lysosomes is important for the degradation of and elimination of microorganisms that can cause damage in our body. So that is the importance of lysosomes. There are so many microorganisms that can cause disease or damage in our body. So it is these lysosomes that try to eliminate all those foreign microorganisms that can cause disease. Okay, Osiale, thank you very much. Welcome to the YouTube channel. Welcome for joining as a member to the YouTube channel. So that is actually the functions of lysosomes. And then the next one is ribosomes. Yes, ribosome is involved in protein synthesis. Excellent. So you see, number one, when the ribosome synthesize protein, then rough endoplasmic reticulum will now the rough endoplasmic reticulum will then actually uh, package it and transport it and even modify it. That is the functions of the endoplasmic reticulum. Dada, thank you. Yes, but you didn't join. So there are videos that you didn't watch. So try to join and watch the videos, especially the summary video of this course before tomorrow if you are writing the exam tomorrow. So let me actually let's look at another question. So we are done with 2A. So then 2B. So I hope you can still see the question. No, I first 2B. No, no, no. Let me go back. Okay. So let's look at the question 2B. So question 2B says differentiate between receptor mediated endocytosis and phagocytosis. So what is the difference between endocytosis and phagocytosis? What is the difference between endocytosis and phagocytosis, ladies and gentlemen? Who can answer it, please? Our endocytosis, Deborah Joseph, endocytosis is the movement of particles into the cell with the aid of energy. Are you sure? The question says, what is the difference between receptor-mediated endocytosis and phagocytosis? Good evening, sir. I subscribe, but you recent lectures, I don't see them. Yeah, I saw that Ozoji, you joined. You are a member. But if you didn't watch the video, maybe if you didn't see them, you can talk to me, or I would just send those videos after this lecture so that you can watch them. Phagocytosis is the digestion of microbes. Yes, Joseph Deborah. Phagocytosis is the process in which a foreign microorganisms will be engulfed by a cell. So the foreign microorganisms can be any microorganisms that can cause damage. So phagocytosis is a process in which microorganisms are engulfed and get digested by the lysosomes of the cells that engulf them. Okay, someone is asking, please, sir, how can I join the community? Yeah, if you check the YouTube channel, you can see join. So you click on the join, and then you use your ATM cards to continue with the subscription. It's just 750 naira. Okay, Ilya Pamon Ilya says endocytosis is the movement of molecules, vesicles into the cell. Yes, excellent. So you see, Deborah, Joseph, you answer phagocytosis, and then Ilya answer endocytosis. So you see, Otata, endocytosis is a movement of substances or molecules from exterior, excellent, from exterior to the interior of the cell, excellent. That is endocytosis. While phagocytosis is the engulfment of microorganisms by the cells and get eliminated by the lysosomes of that particular cell. So that is actually the answer to that question. So let's move to question 2C. Write Michaelis-Menten equation and explain each of the terms. So please, who can write Michaelis-Menten equation here? I want someone to write it here for me. Who can write the Michaelis-Menten equation, please? Write it. Write the Michaelis-Menten equation. Let me see it. Excellent, Ilya. V is equal to V max plus S. You forget to add S. Plus S all over Km plus S. Yes, Ilya, you need to correct the Vmax. So after Vmax is plus S, then divided by Km plus S. So write it again. Correct it. Let me see. I'm waiting for you to correct it. Yes, excellent. V is equal to Vmax plus S divided by Km plus S. So now, what is the meaning of V? The term V stand for what? Excellent. Also, another person have wrote it again. What is the meaning of each of the term? V stand for what? V stand for what? So you don't know the meaning, so let me say it. The small letter V here, the first V here, stand for velocity. Or you can say rate of velocity. You see, what actually happened, ladies and gentlemen, you know that in enzymology, the, enz the enzymes usually speed the rate of the reaction. So when you have a substrate, so the enzymes will now react with the substrate in order to speed the rate of the reaction. So generally, as the concentration of the substrate and the enzymes increase, then the velocity will also increase. Yes, okay, Orido, how can I join? If you check the YouTube channel, you are going to see um join. So click on the join. If you have ATM card, check the back of your ATM card. You have some numbers, then you input them there, then it will automatically join. It will give you access, or you will join the YouTube community. So the velocity V, it is actually the speed of the reaction by the enzyme. So generally, if you have a substrate, the substrate will react with the enzymes, and then the velocity will increase. So as the velocity increase, it will reach to a stage where the active site of the enzymes where the substrate binds will be completely occupied with the substrate. So it will reach to a level where there is no available active site for the substrate to bind. And that is when the maximum velocity will attain. That is Vmax. So Vmax stands for maximum velocity. That is when the velocity is at peak. Okay, so V stand for velocity, Vmax stand for maximum velocity, and then the next one is Km. What is the meaning of Km? Who can tell me the answer? What is the meaning of Km? What is the meaning of Km? What is the meaning of Km? No, 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 it's not kilometer. It's not kilometer. Hey, no. What is the meaning of Km? No, it's not kilogram. Hey, kilo. Ah, kilo de. No, 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 no. So, hi, kilowatt. No, I don't know. It's okay. So let me say it is Michaelis-Menten constant. Michaelis-Menten constant. So write it. Michaelis-Menten constant. Write it. Let me see. Michaelis-Menten constant. Hey, Osumui. Oh, sorry, Osamui. You are laughing. Michaelis-Menten constant. Michaelis-Menten. Let me write it. Wow, this Rofiat I have already no is you write Michaelins no. So look at the correct spelling Michaelis-Menten constant. And what is the meaning of Km? Km is it measures the affinity of the enzymes toward the substrate. It measures the affinity of the enzymes toward the substrate. So the lower the Km value, then the higher the affinity of the enzymes toward the substrate. Let's say you have two enzymes, you have enzymes A and enzymes B. And in enzymes A, you have a Km of 2 milligram per mil, let's say 2 milligram per mil, and enzymes B have 1 milligram per mil. Which one have the lower Km value is the enzymes B. So that means that the enzymes B have more enzymes activity or have more affinity of the substrate. It means that it can easily convert the substrate to products because of its lower Km value. So the lower the Km value, the higher the enzymatic activity. So then, so we know the Km. Then the next one is S. Who can tell me what is S? What is S? No, it's not second. It's not second. It's not speed. It is substrate. You see, that is why I'm telling you this. Please, ladies and gentlemen, I will advise you so that you will avoid most of these kind of problems. I gave you a video last time that the best way to pass most of your exams is to be reading every day. So that is why I'm thinking I'm just advising, if possible, let's try as much as possible to start doing tutorial on time. So I might not be actually your tutor, but we can still make use of this YouTube channel for your general study. So what we are going to do, we are going to maintain that group. I talked with the admin. At least we will be actually using this YouTube for the general studies. So that is why I asked you to try. I told you to try to join the YouTube community so that we'll be using the YouTube to be doing a general tutorial and also answering past question paper. So that is why I encourage you to try and join the YouTube community. Okay, thank you. So the S stand for substrate. So let's actually look at question 3A. Question 3A. A dietitian in your hospital has analyzed food samples A and B, where he reported their glycemic index to be 65 and 23 respectively. As a medical practitioner, suggests with reasons which of the food substances you will avoid administering to the diabetic patients in your ward. So, what is the answer to this question? Ladies and gentlemen, answer this. Why is it that it is sample A? Yeah, excellent. If you check the formula very well, you will see a concentration bracket. So you can call it substrate concentration. So you see a glycemic index is an index that try. Okay, are you not seeing the question? Are you seeing the questions or you are not seeing the question? Please, I'm asking, are you seeing the question? Okay. So now you see the question says a dietitian in a hospital has analyzed food samples. We have food samples A and food samples B. So samples A have a glycemic index of 65, and then we have a sample B has a glycemic index of 23. So as a medical practitioner suggests with reasons which of the samples you will avoid administering to the diabetic patients in your ward. So you see, what I wanted to say here, ladies and gentlemen, uh, glycemic index is an index that tries to measure the effects of a food substances on rising the blood sugar. So therefore, the one that have the high glycemic index, it means it is more danger. It is more danger to diabetic patient. So therefore, the answer here is A, because A has higher glycemic index, which can actually, uh, which might actually cause the high blood sugar. So therefore, it should be avoided. So therefore, the answer is A. And the reason is because it has high glycemic index, which have a greater chances of causing or of increasing the crisis of diabetes. Is that clear? Is that clear? Do you understand, please? I'm waiting for your response. Okay. So now, question 3B. Explain why humans cannot successfully digest cellulose. Who can tell me what is the reason why human we cannot digest cellulose? Remember, cellulose is one of the examples of homopolysaccharide that is found in human. So, but now, why is it that as humans, sorry, that is found in plant, not in human? We found them in plants. But now the question is, why is it that we as human we cannot digest cellulose in our body? Who can tell me what is the reason? Why is it that we cannot digest cellulose as human? I'm waiting for your answer. It is because it can't be dissolved in water. No, no, Deborah. Deborah, you look like excellent student, but you didn't join the YouTube community. So it means that next time we are not with you for metabolism, general physiology, and general microbiology. So why is it that we cannot digest cellulose? Excellent, Ilya. It's because we don't have the enzymes to digest it. So what is the name of the enzymes? What is the name of the enzyme? Because human don't have the enzyme that can digest it. So what is the name of the enzyme? Zaytuna. What is the name of the enzyme? Excellent, because they lack cellulase. So cellulase is the enzymes that digest cellulose. So it doesn't have that enzymes, and that is why we cannot digest cellulose. Rather, there are some microorganisms that we have them in our body. We call them intestinal microorganisms. So they are the one that assist us. They are the one that assist us in digesting the carbohydrates, which is the cellulose. So now let's move to question 3C. Let's look at the question 3C. List three proteoglycans that form the major component of connective tissues in humans. So what are the proteoglycans that form the major component of connective tissues in humans? So who can give me the answer to these proteoglycans? I'm waiting for your answer. What are the proteoglycans that form the major component of connective tissues in humans? I'm waiting for your answer. You don't know it. So who knows it? The major proteoglycans that are found in human. In the formation of connective tissues in human, we have also the corine. The corine, let me write it here, the chondroitin, chondroitin sulfates. That is number one. And then number two, we have the corine. So these are actually the examples of proteoglycans that are found in humans. We also have agrican. Excellent, excellent. So we have agrican. So these are the three actually examples of the proteoglycans that form the connective tissues in human. Yes, excellent. So let's look at question 4. You see, question 4A is one of the most simplest questions that you are going to have. So who can tell me what are the four differences between DNA and RNA? Please, who can say it? Please, I need answer. What is the difference between, what are the four differences between DNA and RNA? Yes, agrican, versican, and the Korean here. So now the next question is, yeah, what the oxy? Yeah, but the question now is, what is the difference between the deoxyribonucleic acid and the ribonucleic acid? What are the differences? Four differences between them. What differentiate them? So let me say it. DNA is single-stranded while RNA is double-stranded. DNA is double-stranded, RNA is single-stranded. That is one. The sugar in DNA is deoxyribose, while the sugar in RNA is ribose sugar. Then another difference, so I mentioned two difference. Then the nucleotides or the heterogeneous bases that are found in DNA, there is thymine, which is actually in RNA, is replaced with uracil. So in RNA, remember that we have four bases in DNA, and the four bases that are found in DNA, we have guanine, we have adenine, we have cytosine, and we have thymine. Then in RNA, we have cytosine, and then we have uracil instead of thymine. So you see, these are the three differences. And then the fourth difference between DNA and RNA, the DNA is found in the nucleus, while RNA can be found in nucleus and the cytoplasm. DNA is more stable while RNA is less stable. So these are the four major differences between DNA and RNA. So please write them down. Now I don't need to write this. You can actually come back, because we shouldn't be wasting time. It is already getting late. So you can actually come back anytime and watch the video so that you can get the answer. But let me just repeat it. Number one, DNA is double stranded, RNA is single stranded. The sugar in RNA is ribose, while the sugar in DNA is deoxyribose. And then in DNA, we have thymine, and then in RNA, the thymine is replaced with uracil. And then DNA is more stable, while RNA is less stable than RNA. And then, so let's move to question 4b. The central dogma of molecular biology involves three distinct processes. Describe each of them. So when we say central dogma of molecular biology, it means how the DNA converted to RNA and how RNA converted to protein. So these distinct processes in which DNA convert itself to another molecule of DNA is called replication. It's called replication. So that is one of the process, that is replication. And then the second process is when the RNA converted, or sorry, is when the DNA converted into RNA, and the process is called transcription. And then the RNA is converted into protein, and then the process is called translation. So let me write it here. We have replication, one, we have transcription, and then last one we have translation. So these are the central dogma of molecular biology. So these are the processes. So the first one, replication, is when the DNA copy itself into another molecule of DNA, and then transcription is when DNA copy itself into RNA, and then translate concept of central dogma of molecular biology. Is that clear? Is that clear? I'm waiting for your response. Okay. So then the next question, which is question 4C, describe the mechanism of action of azidothymidine as an antiretroviral drug. So antiretroviral drugs, retrovirus is a virus that causes Chidema, please sir, I'm not clear on this. The question says we should actually describe the central dogma of molecular biology. There are three processes, so we should describe them and explain them each. So I said that we have replication, transcription, and translation. So Chidema, when DNA convert itself to another molecule of DNA is called replication. And then when the DNA converted into RNA is called transcription. And then when the RNA molecules translate into proteins or convert or translate RNA into protein, then the process is called translation. So these are the three processes that involve in the central dogma of molecular biology. So briefly now, question 5, state one protein associated with each of the following. Transmission of nerve impulse. So the answer is neurotransmitter. Question 1, question B, Roman figure 2, ion storage. The protein that actually involved in storing protein is called, that involved in the storing ion, Fe, is ferritin. And then the proteins that actually involve in immune protection is antibodies. Then question C, describe, okay, I forget, question 5A, describe the levels of organization of protein structures using insulin as a model. So the organization of proteins actually organize itself in three levels. We have primary primary structure, we have secondary structure of the proteins, we have tertiary structure of the proteins, and then lastly we have quaternary structure of the protein. So insulin is a protein, so therefore it has four levels of organization. It can be number one as a primary structure. So what describes the primary structure of protein? What determines the primary structure of protein is number one, the peptide chain. You know that there is a peptide bond. So a peptide bond is a bond that joins the amino acid sequence. So the peptide bond is a bond that actually leads to the primary structure of protein. While the non-covalent interaction between or within the primary structure, and this non-covalent interaction, it can be hydrogen bonding, hydrophobic interaction, and also electrostatic force of attraction are the examples of non-covalent forces. So when you have these non-covalent forces within the primary structure of the protein, that is actually what describes the secondary structure of the protein. But when you have this non-covalent interaction between two polypeptide chain, that is when you have the tertiary structure of the protein. But when you have multiple polypeptides joined together with the non-covalent interaction, that is actually what leads to the quaternary structure of the protein. So I think now I answer question 5A. I answer question 5B. Transmission of nerve impulse protein that actually involve in the transmission of nerve impulse is neurotransmitter. Ion storage, we have ferritin. And then Roman figure 3, immune protection. We have antibodies. Then question 5C, describe the biochemical basis for sickle cell anemia. Biochemical basis for the sickle cell anemia. So generally, the proteins that is affected, which actually lead to the sickle cell anemia, is hemoglobin. So hemoglobin is a protein that has two chain. That is, that has both alpha chain and beta chain. So it has two alpha chain and two beta chain. So in beta chain, one of the beta chain contain 141 amino acids. And at position 6 of that amino acid, there is an at position 6 of the protein, there is an amino acid called glutamic amino acid. And glutamic amino acid is a polar amino acid. But due to mutation and some problem, the glutamic acid is replaced with valine. And that is what lead to the change of the shape of the red blood cells, leading to the sickle shape. So it means that the story and the narration of sickle cell anemia is just due to a change of one amino acid at position 6, and the amino acid is glutamic acid that is replaced by valine, and that is what changed the story. So this is the biochemical basis for the sickle cell anemia. That is the change of glutamic acid with valine at position 6 of the hemoglobin, and that is the answer. Then question 6A. Explain why polyunsaturated fatty acids are essential in humans. They are essential in humans because we don't have the enzymes that can synthesize polyunsaturated fatty acids. So examples of this polyunsaturated fatty acid, we have omega-6 fatty acid, we have omega-3 fatty acid. So they are fully unsaturated, meaning that they have multiple double bond or triple bond in them. So we cannot synthesize them. We don't have enzymes machinery that synthesize them. So that is why they are essential. So we need to get them from our diet. Then question 6B, why or explain why triacyl glycerol are more efficient means of energy storage in humans than carbohydrates. The answer is, if you check the triacyl glycerol, it has fatty acid, it has three fatty acid and one molecules of glycerol. So in the fatty acid chain, there are so many hydrogen. So it is highly reduced because it has many hydrogen. The fatty acid have many hydrogen in them. So it means that they are highly reduced. And it is because of that reduced properties of the fatty acid that is why lipids or triglycerol produce energy more than carbohydrate. So the answer is because fatty acid is highly reduced. It is highly reduced. That is why it produces energy more than carbohydrates. Then question 60. Describe the functions of prostaglandins. So who can tell me what is the functions of prostaglandins? The functions of prostaglandins. What are the functions of prostaglandins? You are too silent. Are you tired? Prostaglandins. No, sir, we are not tired. Okay. So, what are the functions of prostaglandins? What are the functions of prostaglandins? They play a role in uterine contraction. Excellent. So, during labor, these prostaglandins are help in the contraction and the relaxation of the uterus. Excellent. Then, what again? It causes pain like headache. So it is one of the functions of prostaglandins. Then which other function? So one, it causes uterine contraction. Two, it causes pain. And then third, it helps in menstruation. It helps in, it causes inflammation and also protect mucosal lining of the stomach. Excellent. Chimeria. It also assists or helps in menstruation. It can also act on hypothalamus to cause fever. So it causes fever, it causes pain, it causes inflammation, it causes uterine contraction, it regulates kidney function. So these are actually some of the functions of prostaglandins. So let me see, I think this is actually the last one. Yes. So this is actually the last question from this question paper. So we are done with this question paper in 1 hour 25 minutes. So ladies and gentlemen, I think we have actually achieved something very important. So I will advise you to, yes, thank you so much. Thank you too. So I always encourage you, if you know that you didn't join the YouTube community, try to join it now, and I'm going to send you the link for the summarized video. So thank you very much. So we are going to uh we are done. I think we are going to stop here. So any question? Do you have any question? Akinde Fola, thank you very Akinde Fola. Thank you very much for joining the YouTube community. So we have so many members now in the channel. So if you know that you didn't join, try to join the YouTube community. It will actually going to be of help to all of you. So thank you very much. I think we are going to end it now, unless if you have any question. And if there is no question, then we are going to end the live. So no question. Yes, okay. Ibera Lillian, you okay? I will now. You can send. I will check the YouTube channel. You can see join, and if you didn't see join, then maybe you can talk to me privately. I will send you the link. Yeah, or let me just send the link to the group. So for those of you that wanted to join, I will send it. Thank you, sir. Please, we are waiting for the revision link. Yes, I will send it to the group now, Chidimma. So thank you very much. Bye bye. Thank you very much. I'm going to end the live now till we meet next time. Thank you very much. Bye bye. Okay, I have been trying to join, but I could not. Yes, I will send you the link now to join in the group. So thank you. Bye bye.